Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. This series of videos that I'm recording are intended for use by students that are enrolled in my human anatomy and physiology classes. Now this particular video is intended for the students in my Physiology 2 class, Part 2 Human A&P, and this is the fourth video in a series of sensory physiology videos. We're going to be covering the special senses, or at least beginning them in this video. I'm trying to keep the videos to about 30 minutes or less, because it becomes hard to follow along after a while, and it's very difficult to get them to load in a timely fashion into YouTube. So I'm using some crude equipment, as you guys are aware of from some previous lectures I've talked about. I'm not a technologically savvy guy. I'm doing this on a marker board in my dining room with my cell phone, and this is what we got. That's because we're in this coronavirus or COVID-19 shutdown, and we've had to switch from face-to-face to online literally overnight. So anyone else out there in YouTube land, if you stumble across this and you find it helpful, awesome. Use it. Maybe it'll help you understand some of the complex physiology. Now, if you're in my class, we're going to be following along on the note set on page 8 where it says olfaction. Olfaction is the sense of smell. It's considered to be the oldest of the special senses to evolve. That's how ancient organisms in the ocean would follow a concentration gradient of chemicals to find a food source or avoid predators and find mates as well. The olfactory receptor cells are located in a layer of tissue in the nasal cavities and the paranasal sinuses called the olfactory epithelium. So when we look at this tissue now, what I'm going to draw out is going to be very similar to what you're going to see on the slide for lab for this. And it's going to help us understand the sense of olfaction and how olfactory physiology works. And we're going to get into a little bit of what we call signal transduction, one of my favorite topics and something that I studied in graduate school. So the olfactory organs, as I mentioned, are in the nasal cavity and the paranasal sinuses, and it is a massively folded layer of tissue. Now, as you've probably become aware in physiology, anytime I have an area, if I can take the same distance, but I can fold up the membrane, then I actually have a lot more surface area in the same distance. So everywhere that we see folds in physiology and anatomy is to increase the surface area. The more surface area, the more actual olfactory receptors I can fit in there, and that increases the likelihood that a molecule inhaled will bump into a receptor and trigger olfactory transduction. Now, when we look at this epithelium, one of the things, I don't know if you recall this from part 1 A&P or not, but I'm gonna do some drawings to get us sort of oriented here. Let me draw it facing the other direction. Okay. If I had the human skull here, as you remember from part 1, way up here is the nasal bones, and then we have an ethmoid bone. We have nasal, lacrimal, and then the ethmoid bone, which sits inside in between the two orbits. On the ethmoid bone was a little piece that stuck up called the crista galli. And on either side of the crista galli, if I could look down on top of it, there was a little plate of bone that had hundreds of little tiny holes in it. Those holes or openings are to allow axons to come through. What sits on either side on that cribriform plate, that plate on either side of the crista galli, is the olfactory nerve, cranial nerve number 1, sits up here like this on either side. And the axons from the epithelium here are going to run up through the cribriform plate, synapse on the olfactory nerves, and then that's going to send an action potential into our brain. So that's sort of the anatomy that we're looking at. What I'm going to draw is I'm going to draw a mid-sagittal section as if I cut right through all of this, and you're going to see some of these little holes and some bone from a mid-sagittal view. So now, this is sort of the anatomy and the layout of things so that I can go into the physiology of it over here, and we can understand how this stuff works. Now, there's a piece of bone that's going to have some holes in it called the cribriform plate. Sitting above the cribriform plate is cranial nerve number 1 called the olfactory nerve, or the olfactory bulb, because it's kind of bulb shaped at the end. So this is cranial nerve number 1, the Roman numeral 1. It's going to have neurons in here that are going to carry action potentials to the brain for us to interpret the smells that we're detecting. Now, covering the cribriform plate and a lot of the bone is going to be a messy layer of connective tissue. And There's a whole bunch of mess in here. This particular layer of connective tissue is going to be a layer that is called the lamina propria. Orange doesn't show up too well on the cameras. I've got a little monitor here on my laptop that I can see, so if you see me looking down a lot, it's because I'm trying to see where I'm at on the board. So I'm staying somewhat in view of the camera. Anyway, this layer is very often called the lamina propria. It means the proper layer. It's a common theme in the human body. And then the layer sitting on top of that lamina propria, which the lamina propria is some type of connective tissue usually, is going to be a layer of epithelium. So if you recall from histology, when we studied the tissues, epithelial layers, there's 4 major tissues: epithelium, connective, muscle, and neural. Epithelial tissues cover and line the surfaces of the body, like your skin. The epidermis is an epithelial layer, but also the internal cavities. If I reach in and scratch my throat, if you reached in from the other end and scratched, then you would be touching epithelium all the way through. If you swallowed a piece of string and you flossed yourself, you would be touching epithelium all the way through the digestive tract. So, always and forever, right underneath the epithelium, with a rare exception, is a lamina propria, a layer of connective tissue. Now, when we look at the olfactory epithelium, if I can magnify some of the cells are going to take on distinct morphologies and have different names and functions. One of the cells, or most of the cells in this layer, are going to extend axons up into and synapson cells in the olfactory nerve, or the olfactory bulb, okay? These cells that have these little folds on them are going to be called olfactory receptor cells, okay? This is my abbreviation for receptor. Anytime I have an R with a circle, it can either mean right or it means receptor. In this case, these are the olfactory receptor cells. These are neurons that have developed these little hair-like structures that stick off called olfactory cilia. And on those olfactory cilia, there are proteins embedded in them. And those proteins embedded in there are called odorant binding proteins. So if I were to look on one of these cells, there'd be all these little tiny proteins embedded. I don't know if you can see this, but I'm going to magnify and draw this up better for you in just a moment. So the olfactory receptor cells are the cells that actually detect the chemicals that we're smelling in the environment, called odorants. When we inhale an odorant or sniff it in, it will bind to some receptors in the membranes of these olfactory cilia and trigger a cascade of events that results in an action potential or a change in action potential, signaling the cells in the olfactory nerve. This is that specialized receptor cell and the first order neuron, so to speak. Now, mixed in with those olfactory receptor cells, if we look under a microscope, we'll see these other cells embedded in here that look like columnar cells. I'm going to leave a gap in here for a reason, but a lot of these cells that look like little columnar cells in here are going to be called support cells. Now, eventually the support cells are going to develop into new olfactory receptors. Now, when we studied the organelles of a cell in part 1, one of the organelles was called centrioles. As you know, centrioles, these little tube-like proteins, are going to separate chromosomes during mitosis when a cell divides. Without centrioles, the cell can't form a mitotic spindle, it can't separate the chromosomes. Well, as most neurons are maturing, they lose their centrioles. They lose the ability to divide. So when a neuron dies, it's wiped out forever. We can't usually replace neurons because there's no cells to replace them. There are two exceptions, two really well-known exceptions to that rule. One is olfactory receptors. Olfactory receptors live between 7 to 14 days, about 10 days on average, and then they die and are shed, and these olfactory or support cells will start to develop modified dendrites that we call olfactory cilia, and they'll start to grow an axon and rewire. So we're constantly shedding and replacing olfactory epithelium. It's one of only two good known examples of what we call neuronal replacement. So, They also do some other support, but for the most part, they're just going to grow up to become olfactory receptor cells as we replace them. Now, right at the base, at the border between this epithelial layer and the lamina propria, are these other cells that sit here. I'll put them in a different color so that you can see them, but these little red cells are going to be called basal cells. My drawing got a little bit crowded, so I can't point to them up there, but these basal cells live at the base of the olfactory epithelium. So this entire layer here of these three cells, I'll put a little asterisk, that whole layer is called the olfactory epithelium. Now the olfactory epithelium has the olfactory receptors, the support cells, and the basal cells. One thing that's important about this basal cells is that they are mitotic. They divide by mitosis, and when they divide, the old cell stays behind, the new cell starts developing into a support cell, which will grow up to become an olfactory receptor. So that's their importance. Which, by the way, these are considered to be these are considered to be adult stem cells. They're stem cells, cells from which all our organs can stem, ancient cells from the body from which we originally developed in the womb, but they are adult stem cells. And so scientists are actually using these cells and placing them in mice and other organisms that have experienced spinal cord and brain injuries. And since they're already destined to become neurons, they're wiring themselves up as new neurons. And we're actually going to eventually develop the ability to transplant these cells and heal spinal cord injuries and brain injuries. Possibly. There's some hope for that. Anyway, those are the basal cells. Now, all of this is your olfactory epithelium. This is the lamina propria. Two more things I want to show you before I get into the detail of olfactory transduction. Some of the columnar cells develop into what we call goblet cells. Goblet cells are kind of narrow at the base and thicken up out here. And so these goblet cells can secrete mucus. Oops, I didn't spell that right. Excuse me. They secrete a pigmented mucus. That mucus is going to have a number of functions, but it's going to coat the epithelium. And of course, as you're familiar, when the mucus dries, you develop boogers, you know? So these goblet cells are called goblets because if I flipped it upside down, they kind of look like a wine goblet. They're skinny on the bottom and thick on top, or narrow in one area and thicker in the others. They got the name goblet cells. They fill with mucus and then rupture and secrete that mucus. In addition to the goblet cells, in the lamina propria are also olfactory glands. Now, olfactory glands are large mucus glands. When something starts to irritate the olfactory epithelium or the respiratory passages, we can hypersecrete mucus to flush away all the debris or dirt or irritants. And that happens because there are little round glandular cells up here. And sometimes these cells have a duct that will make it to the surface of the body. They do always, but we don't usually see them when we slice it for tissues. But they can secrete more mucus than the goblet cells, and they can flood mucus into the epithelium to wash away debris. So this is kind of the anatomy of the olfactory epithelium, communicating to the first order neuron in the olfactory bulb and the lamina propria. The lamina propria contains mucus glands, Bowman's glands, that can secrete a lot of mucus. Now, before I go into the transduction, I just want to go over the functions of mucus. So I'm going to erase some of this board here. I'm going to erase most of my writing since we've already labeled it, and you can pause your video and watch it or look at it. I'm going to erase all my labels and make some room over here. And I'm going to do the steps of olfactory transduction in just a moment. Okay? But there are four major functions to the mucus. Do I want to list all four? I think so. I might just list three, but anyway. One of the things that the mucus can do is that it can trap odorants. When we inhale air, if you look at the anatomy of the nasal cavity, we have three nasal conchae in here. Those conchae create turbulence. They cause the air as it's blowing in to hit them and swirl around and make all the air brush up against the walls. The walls of the cavity are coated in mucus. Now, odorants, the chemical structure of odorants is such is that in that they will dissolve into the mucus and move through. They get trapped in the mucus and they dissolve through it and bump into the olfactory cilia. So one of the functions of mucus is to help trap odorants, and it increases our sensitivity to those odorants so that they're not just blowing by, but they get trapped and they're forced to diffuse through and bump in. So they trap odorants to increase olfactory sensitivity. Okay? Now, another function is they can trap dirt or debris and wash it away. Since the dirt and debris are not going to dissolve into, or even if they do dissolve into, they don't have the chemical structure to lock into or bind to the receptors, these don't trigger olfactory sensations. So one of the things that happens is these cilia can also beat and push the mucus out and away from the epithelium and force it out of our body. And so they're removing dirt and debris so that that stuff does not become trapped in our lungs, because our respiratory and olfactory sensations are part of the same system. They're part of the respiratory system. And as you inhale dirt and debris, if it clogs up your lungs, obviously you can't breathe. Kind of like if you take an air conditioning filter out of your AC, it's all clogged with cat hair and dirt and dust and debris. All that particulate matter is blowing around in the air that we inhale, and over time your lungs would clog up. We can't throw them away like an AC filter. So this helps protect the respiratory system. So it doesn't necessarily always play a role in olfaction. It helps protect the respiratory system by trapping dirt and debris and washing it away. Really these are the four functions. I listed it as four, but it's kind of really three, because not only do they trap the odorants, but that increases our sensitivity to olfaction. Oh, we could list this one. They protect also by preventing the olfactory epithelium from drying out. Okay, so if I took two wet towels and tossed them on a table, but I put a fan blowing across one of the two towels, the water on the surface of the first towel would be blown away by the air. The water underneath would, by osmosis, move to an area of lesser water concentration, and it would get blown away. And we systematically blow away all the water. So the towel with air blowing across it will dry much more rapidly than the towel without it. Well, as we're inhaling and exhaling, we would dry out our olfactory epithelium and it could crack. Well, the olfactory mucus traps moisture, kind of like putting Vaseline on your skin. It doesn't let anything penetrate, and so it prevents the olfactory epithelium from drying out. So it protects in two ways. It traps dirt and debris and washes it away, and it prevents the olfactory epithelium from being dried out and cracked. So know those functions of olfactory mucus. They're really important. By the way, I am aware that I drift out of screenshot because my camera's only so wide. So I apologize, but I got a face made for radio, so y'all don't want to see me anyway. But nonetheless, all right, I'm going to erase these functions, and now we're going to go over olfactory transduction. Okay, it's not entirely all like this, but this is one of the methods, one of the pathways of olfactory transduction. Well, actually, most of smell is like this, and taste works very much like this. Gustation, or the sense of taste, works very similar to olfaction. So when we understand olfactory transduction, we're also going to understand gustatory transduction. So now, imagine this, okay? I'm going to take this little neuron here, and I'm going to magnify him to some degree. I'm going to show you the steps of olfactory transduction, and I'm going to list them here, and you should know them. Sometimes in anatomy and physiology, students ask, well, why do I need to know all this? I just want to be a nurse. I just want to be an OTA. Sometimes it's not only about can you learn the material, it's who's willing to jump through the hoops and do all the stuff that it takes to get through to the end. Those who are not willing or are not able don't make it. And those who are, it will show that you not only one can learn steps and processes, but you're also willing to jump through whatever hoops you got to jump through in order to become the professional. They're betting on you when they allow you into one of these programs when you get accepted. So learn this stuff. You may never see it again. Some of you might, but just learn it. It's the process. Plus, it's absolutely fascinating. If you're not in love with the human body, something's wrong with you. You're in the wrong major. All right, so look, this olfactory receptor cell. On one end, I'm going to have these olfactory cilia. And it is a lipid bilayer, so I'm going to draw two layers. You know how the cell membrane should look. On all the way on the other side of the neuron, getting close to where the axon hillock and the axon would be, I'm going to have some other stuff over here. Now, embedded in the olfactory cilia are different proteins. Some of these proteins, I'm going to draw them with specific shapes. Okay? All of these proteins can bind to different odorant molecules. The molecules that can bind here are called odorants. And these proteins that are embedded in the cell membrane are called odorant binding proteins. There's many, many, many of them, and they all fit specific odorant molecules. That's why I've given them somewhat of a three-dimensional shape. Now, let's say I have these molecules called odorants. An odorant is any substance that can trigger olfactory transduction. These proteins in the cell membrane are called odorant binding proteins because of their three-dimensional shape and their electrochemical properties. If a certain odorant comes in close proximity to certain ones of these, it'll bind. Another odorant will bind to a different receptor and a different receptor. So if I had odorants that had a three-dimensional shape like a little wedge or a circle or a square, then these odorants would only bind to these proteins, odorant binding proteins. This odorant would only bind to certain odorant binding proteins, and so on and so forth. Okay? Now, when the odorant gets trapped in olfactory mucus, so we'd have a layer of mucus here. As an odorant gets trapped in the mucus and dissolves through, it can bind to an odorant binding protein. Now, one of the things I try to explain in all of my classes, and you'll see me do this several times, is, as understanding the structure of a protein. So I'm going to draw over here just to give you a background idea of what I'm talking about, and then I'll erase it so we can finish this process. When we synthesize proteins at a ribosome, really a protein is just a long chain of amino acids. It's a whole bunch of amino acids stuck together by bonds called peptide bonds into a long chain. There are 20 different amino acids that we can build proteins from. What makes one protein different from another is the number and the order of the amino acids. These amino acids have a series of carbons and hydrogens and oxygens, rare occasions of sulfur and phosphate, sulfur I should say, and they can have little side groups. And let's say these two amino acids have a side group that likes to bind together in a chemical reaction. If I can fold this protein over, and these proteins are like spaghetti floating in fluid, if they get close together, those two proteins will form a chemical bond, and those two amino acids I should say, and the protein begins to fold. Well, that might naturally bring two other amino acids or several other amino acids in close proximity, and over time the protein starts to knot up into a three-dimensional shape. We call that protein folding. If something binds to this protein, let's say there's a series of amino acids right here in this area that are exposed, and another molecule has some other substances that can bind to those. When it binds to those amino acids, it might be me binding something to this one right here. And if there is a higher affinity or desire to bind to that, then it's going to break that chemical bond and the protein can unfold or change shape. So proteins get folded, and certain parts of the protein, if they bind another substance, can unfold one piece of the protein or cause a partial unfolding. They change their three-dimensional shape, so to speak. The reason I tell you that is because that's exactly happens when an odorant binds to an odorant binding protein. Now, odorant binding proteins are actually inactive molecules that can change the physiology of another molecule. So, what happens is this. When an odorant binds to an odorant binding protein, these two odorant binding proteins will change their shape and a part of the molecule might stick out a little bit. That's not exactly what happens, but sort of. Now that that's exposed, it can interact with another molecule. Now, our cells need tons of ATP for energy. So it turns out that molecules of ATP are being pumped out by mitochondria and floating around inside the cell. Now, ATP can be converted into another molecule, and I'm going to explain this in a second. But before we do that, there's a third substance in here that we can worry about. I have the odorant binding protein that's been activated by an odorant binding to it. There's another molecule called adenylate cyclase. Anytime you see ASC at the end of the word, it's an enzyme. Adenylate cyclase is an enzyme that can form a cycle or a ring out of a molecule called adenylate. Adenylate is just another form of adenosine. There is somewhat of a difference, but nonetheless, adenylate cyclase, and I'm going to label adenylate cyclase ADC. This little enzyme, it's a protein with a biochemical function, but it is not functioning inside the cell until an odorant binds to an odorant binding protein. And when this little piece pops out or gets exposed, then the odorant binding protein becomes activated. What that can do is take an inactive form of adenylate cyclase, and if they interact, it can turn adenylate cyclase into an active enzyme. It makes it change shape. Adenylate cyclase is going to take a molecule of ATP and convert it into another molecule called cyclic AMP. So this little substance that I just drew, that is little c, capital A, M, capital P, stands for cyclic AMP, or cyclic adenosine monophosphate. So let me explain something so that you understand this, and then we'll go through all the steps. If I take ATP, which is basically a double ring molecule called adenosine with 3 phosphates in a row attached. If I break off one of the phosphates, I have ADP, adenosine diphosphate. If I break off a second phosphate, I have AMP, adenosine monophosphate. The phosphate wants to have a little bond right here. This enzyme, adenylate cyclase, can actually reattach this back to the adenosine molecule. So now I have my phosphate bound in a little cycle or ring. Instead of a straight line, it curves it around. So it really rips off 2 phosphates and bends the other one around and makes a ring out of this adenylate molecule with a single phosphate. So we call this cyclic AMP. The beauty of that is when I convert ATP into cyclic AMP, this molecule in its new shape can go and bind to ion channels, chemically gated ion channels that are on the opposite side of the cell, where the odorants can't get to. And when cyclic AMP diffuses over and binds to this, it opens, say for example, maybe a sodium channel. Now my sodium ions, with their charge, can flood into the neuron, and if they depolarize it to threshold, it fires an action potential. That action potential will run through the axon into the olfactory nerve, and then possibly stimulate the next neuron to fire an action potential. So, the series of steps in the transduction of this cascade is as such. I'm going to put some numbers on the diagram. I'm going to erase all of this, and I'm going to put the numbers in order so that you can, go through the steps and understand them. In the first step, we're going to have the odorant dissolve through the olfactory mucus and bind to the odorant binding protein. In the second step, we're going to activate the odorant binding protein. In the third step, or actually, that's all really one step. The odorant dissolves through the olfactory mucus, it binds to the odorant binding protein and activates it. The activated odorant binding protein will convert adenylate cyclase into an active form. When that enzyme is activated, that enzyme can then convert ATP into cyclic AMP. Cyclic AMP will bind to the ion channel, altering the membrane potential, the resting membrane potential, and we can fire an action potential off into the brain, or at least into the olfactory nerve, and signal olfactory transduction. Those are the steps involved in this process. Okay? It's a complex signaling pathway. It would be much simpler if this ion channel was over here, and we just bound an odorant to it. So one has to ask, why do we have this complex signaling pathway? Why not just have the odorant binding protein be an ion channel? Kind of like, acetylcholine and acetylcholine receptors. And the reason is this. One ion channel opening will only allow a small amount of ions in before we break down the neurotransmitter or the chemical. That would never cause us to reach threshold. We would have to flood this with millions of molecules of an odorant to open enough ion channels to signal the brain to cause an action potential, to get that 10 millivolt change from resting membrane potential to threshold. But in this system, I can have one or two odorant molecules bind to odorant binding proteins. When an odorant binding protein is activated, one odorant binding protein, in a fraction of a second, can give me over 1,000 adenylate cyclase molecules. Now I've amplified the signal to be 1,000 times stronger. Every one of those 1,000 of these can give me about 1,000 cyclic AMPs. So if every step of the way I get 1,000-fold amplification, by the time I get to here, I can open 1,000,000 ion channels in a fraction of a second. So it makes the sense of smell much more sensitive. It only takes a couple of odorant or a few odorant molecules to give us a strong enough signal, because this pathway amplifies the signal 1,000 times in each step, from step 3 to step 4, so that I open 1,000,000 ion channels. It increases olfactory sensitivity. So there's a question in the note set that asks this. Anyway, let's go through the steps. Now I know in my note set I only listed 4 steps, but I've expanded it a little bit. That's exactly what's happening. An odorant dissolves through the mucus and binds to an odorant binding protein in the membrane, activating it. That causes adenylate cyclase to become activated. Adenylate cyclase converts ATP into cyclic AMP. That will bind to ion channels near the axon and alter resting membrane potential. So let me erase all this. Let's write these steps out so that you can see them happening. And then we'll be done with the sense of olfaction for now. I'll stop the video and we'll do gustation and some other stuff later. So here's the steps in olfactory transduction, okay? And I like to ask questions like, please put these steps in the correct order, etc. So you should know the steps of olfactory transduction in order. In the first step, an odorant dissolves through the olfactory mucus. And binds to an odorant binding protein. I'm just going to put OBP for odorant binding protein. The odorant binding protein is activated. This activates,